Hi guys, my name is Michaela Rose and I'll be presenting over plague. So the first plague was in the 6th century AD and it killed an estimated of 100 million people. The last great pandemic occurred in the late 1800s and was transmitted across the world, primarily by rat-infested ships. Signs and symptoms. The three manifestations of infection occurred with the bacterium causing plague were one, the pneumonic plague, which was a respiratory disease. It caused headaches, pain, chest pains, fever, cough, pain and difficulty breathing, and appearance of paleness. Bubonic plague, the bacterium injected by the bite of a flea enters the lymph and is filtered by a local lymph node. Inflammation and necrosis of the node resulting in a swollen lesion called a bubo, usually in their groin or axilla area. These symptoms were fevers, chills, headaches, nausea, weakness, and tenderness of the bubo. Septicemic plague, these cases often progress to massive bacterial growth in the blood. Bacteria in the blood resulted in a disseminated intravascular coagulation, subcutaneous hemorrhage, and a purpura that may degenerate into necrosis and gangrene. Now, because of the darkening of the skin, the plague was often called Black Death, which you can see the darkening of the fingers to your right. The mortality rates at this point were 30 to 50% with treatment and 100% without. Causative agents. It was caused by a tiny, harmless-looking gram-negative rod, Yersinia pestis, which was a member of the Enterobacteria. Pathogenesis and virulence vectors. Only 3 to 50 bacteria cells were required to initiate a plague infection. Y. pestis carries a gene that helps it cause disease in mice and to survive in the flea vector. These genes included a gene for capsule formation and a gene for plasmogen activation, which led to clotting that helped microbes resist phagocytosis, which you can see the little safety pin appearance of the plague. Transmission and epidemiology. Biological vectors are fleas. After a flea ingests a blood meal from its affected animal, the bacteria multiply in its gut. The esophagus becomes blocked due to coagulation factors produced by the pathogen. Being unable to feed properly, the flea jumps from animal to animal to get nourishment. During this process, regurgitated infectious material is inoculated into the bite wound. Plague is amongst the most complex of diseases. It involves several different vertebrate hosts and flea vectors. The exact cycle varies from one region to another. The plague is endemic to the southwestern USA with less than 10 cases per year. I also have a little picture of how it can go through wild animals and people. Diagnosis of infection. Because death can occur as quickly as 2 to 4 days after appearance of symptoms, prompt diagnosis and treatment are imperative. Culture of the organism is definitive and a definitive method of diagnosis. Sorry, guys. Today, rapid genomic and immunochromatographic tests have been developed to quickly diagnose infection. Prevention and treatment. Streptomycin and gentamicin can be used in the treatment, but the threat of drug resistance in Y. pestis could make drugs ineffective in the future. Prevention can include flea and animal control, and vaccines are available for high-risk individuals, such as lab workers that work with Y. pestis. All right, guys, thank you for listening. Have a great day.